Hi all, welcome back to my channel. I hope you guys are doing good. Today I am going to discuss about sequence of blood sampling, that is order of collecting blood samples. As you all know, blood sampling is a common procedure nowadays which helps to diagnose a specific disease condition. It also reveals the functional ability of the organs in our body and also which represents how effective the treatment is. Let's move on to the topic. Phlebotomy. Phlebotomy or venipuncture is a technique in which a needle is temporarily inserted into a vein to provide venous access for venous blood sampling. Before moving into the topic, you should know something about blood collection tubes. As you all know that blood collection tubes are used to collect blood samples which are available in different colors. Most blood collection tubes contain an additive that either accelerates clotting of the blood, that is clot activator, or prevents the blood from clotting, that is anticoagulant. So a tube that contains a clot activator will produce a serum sample when the blood is separated by centrifugation, and a tube that contains an anticoagulant will produce a plasma sample after centrifugation. You should know about this term too, inversion. That is nothing, just how to mix the tube with blood sample. So to achieve the proper mix of additive and blood, each tube must be gently inverted as it is removed from the holder. As I said, all the blood tubes contains some additives. So soon after taking the blood sample, in order to achieve the proper mix of the additives and blood, you have to just mix the tube as it is shown in the picture. So after taking the sample, just make the tube downwards, then turn it to upwards. That is equal to 1 inversion. And each tube requires different times of inversion. We will discuss about that in the further slides. For instance, if a patient got admitted in a hospital and the doctor advised you to send some blood samples, which includes blood culture, CBC, biochemistry, coagulation profile, and so on. So in this case, you have to take the sample according to the order in order to prevent any value changes in the blood reports. So let's see about that. So the first one is blood culture. As it is a sterile procedure, always you have to give first priority to the blood culture. Okay, so after taking the sample, you have to mix that 8 to 10 times, and the additives present in the blood culture bottle is sodium polyanethol sulfonate. Shortly, we can say SPS. The second one is citrate tubes, which is available in blue colors as you can see in the picture. So after taking the sample, you have to mix it for 3 to 4 times, and the additive present in citrate tube is 3.2% sodium citrate. The third one is plain tube, which will be in red color as you can see in the picture. So after taking the blood sample, you have to mix it for 5 times, and it is a serum tube with or without clot activator or gel. The fourth one is serum tube, which will be in yellow color as you can see in the picture. So after taking the blood sample, you have to mix it for 5 times, and this is also a serum tube with or without clot activator or gel. The fifth one is heparin tube, which will be in green colors as you can see in the picture. So after taking the blood sample, just mix it for 8 to 10 times, and the additives present in heparin tube is sodium or lithium heparin. The sixth one is EDTA tube, which will be in lavender color. As you can see in the picture, after taking the blood sample, you have to mix it for 8 to 10 times, and the additive present inside this is potassium EDTA, and the full form of EDTA is ethylene diamine tetraacetic acid. The last one is fluoride tube, which will be in gray color, as you can see in the picture. So after collecting the blood samples, you have to mix it for 8 to 10 times, and the additive in fluoride tube is sodium fluoride and sodium or potassium oxalate. Here we can see order of sample draw. For instance, if your doctor advises you to send only one sample, then you can directly take the sample and send for investigation. In case if he ordered to send multiple samples, then you have to follow this order. Here is an acronym just to remember the sequence. Boys like red and yellow, girls like gray. B stands for blood culture and blue. So if blood culture is there in that multiple samples, first you have to send the blood culture, followed by citrate vial, that is blue. Then red vial, yellow, girls, G for green. So heparin vial, like L for lavender, followed by lavender vial, and the last one is gray, that is gray vial. So one more thing you can notice here, it is very easy to remember how many times you need to mix the tube. So look here, boys like red and yellow. So blue tube, red and yellow tube you can mix for 5 times. And girls like gray, green tube, lavender tube and gray tube for 8 to 10 times. Thank you so much for watching this video. This is very simple to remember. Just keep in your mind, boys like red and yellow, girls like gray, so that when it comes to multiple sampling, you can easily take the sample in that order. I hope this would be helpful for you guys. If you feel the same, kindly like, share this to our friends and subscribe my channel. My channel name is Jebin Shine. Don't forget to press the bell icon button so that you will get the notifications whenever I am uploading new videos. Thank you all once again. See you all again with one more interesting topic. Until then, it's bye from Jebin. Bye bye. Take care.